Hello and welcome. In this section of videos, we're going to cover the following machine learning topics. We're first going to introduce some major classes of machine learning, if you will, and then we're going to focus in our attention on unsupervised learning topics. Those of clustering, that's finding groupings in our data, and then the increasingly essential topics in bioscience research generally of dimensionality reduction, where again we want to reveal underlying patterns or groupings or structure in our data, if you will. But these things are critically important because, of course, our data is getting larger and more complex all the time, where we're measuring many things many times, such as many gene or protein entities over many samples or conditions. That is, we have multivariate data. We're measuring multiple variables over multiple conditions, and we're going to need these approaches to really try and help us make sense of what can be going on in our data sets. In our hands-on section this week, we're going to take these approaches and combine them and explore them in quite some detail and apply them to a real cancer cell classification project, where in this case we're actually going to take data from breast cancer biopsy samples to see what we can learn by applying these machine learning methods. So as I mentioned, we're focusing in on unsupervised learning, where the goal again is finding structure in what we call unlabeled data. That is, we don't know necessarily the labels or the answers for what class or what grouping or the feature that we want to classify. We don't know the answer beforehand. That's typically what we're working with in biology research. And this is in contrast to so-called supervised learning, where we are making predictions based on training, typically with a subset of that labeled data, and then using the model that we developed from that approach and applying it on test data that was not in the training data set. This can include things like predictions from regression or classification models, decision trees, random forests, neural nets, deep learning, these kind of approaches. So these are sometimes used in biology. I don't want to say they're not. We're using them more and more as we get more and more data that is large enough to use these methods productively on. But really we focus in on unsupervised learning. So the third major class that I'll mention just in passing here, it's not typically used thus far very frequently in biology. It's known as reinforcement learning. It is a major area of research, of course, in computer science and machine learning generally. And again, this is because we don't typically know the answer in advance in biology. These techniques, they typically learn in a kind of interactive environment by so-called trial and error using a feedback mechanism. Often folks refer to this as like a reward and punishment approach, with the goal of finding some sort of suitable action model that would maximize the cumulative reward, for example. Basically making decisions based on past experiences. So we're going to focus in here on supervised learning, where again the goal is to find similarities and differences between, for example, data points within our data sets to help reveal structure indicative of some underlying biology that we're researching here. And this is always usually applied to unlabeled data, where we don't know the answers beforehand, what the structure should be, for example. So first up, we're going to delve into Kmeans clustering. So the Kmeans algorithm here, what it does is it breaks our data into k predefined numbers of groups or clusters. Now, with Kmeans, you have to define k at the outset. You have to tell it the number of clusters you want, and you'll see that this can be a limiting factor in this approach that we'll come back to in a little bit. So imagine for a moment that you had some data that we could plot very simply along a line, right? A single number line, a single dimensional of data here. And you knew you had to put them into k equals 3 clusters. For example, your data came from 3 types of tumor cells, let's imagine. So your eyes could probably clearly see that there might be 3 natural groupings in the data that I've drawn here. And I'm showing these with the color here on our data points. So how does Kmeans attempt to define this grouping? Let's look at the steps as we go through. So the first step is you tell Kmeans the number of clusters. You tell it k. We say k equals 3. So we pick, or Kmeans picks 3 distant data points at random and uses these as the initial clusters here. And again, I'm showing these with 3 distinct colors here on the graphic behind me. So the next step is we're going to go to our first unassigned point, the gray point here, and we're going to try and put it into one of these 3 groupings by measuring the distance to those cluster representatives. And I'm showing this with the little colored lines here to each cluster representative. So clearly the blue cluster representative is the closest in this case to this first gray unassigned point. So we're going to assign that to the nearest cluster, in this case blue, and I'm going to color it up blue. And then what we do is we update our cluster representative point. We're going to calculate a new mean value for that blue cluster that includes this new point, and I'm showing that here with the blue line or segment here. And then we move on to our next unassigned point, our next gray point. And again, we do the same thing. We see that this one is, in this case, closest to the green cluster representative in terms of distance. So we're going to assign that to the green cluster and update our cluster representative again to the center here for the next distance calculation that we're going to do. And then we go on. We take the third point, measure the similarity again, and proceed in this way until we have all our points assigned to one of these k=3 groupings. Hold on here a minute. So the k-means result that we just got, that's the one shown on the top here, doesn't look as good just by eye as the one that we were able to come up with just by looking ourselves by eye. That's the top versus the bottom here. So what do we do? Well, first we have to say, well, how do we assess the quality? We have to put a number on this thing. How good is this result? How good is this clustering answer? And what k-means does in this step is it keeps track of the result that we got by the total variance. It puts this score, the total variance within a cluster, the total variation within each cluster, and scores this sum value here that I'm showing by the horizontal colored lines here. It takes this, sums it up, and keeps track of this value, and then tries again within a different initial starting point. Picks another 3 random points that are different than the last one and tries again, does the whole procedure again. So basically we go back to the beginning and do all those steps over again by picking new points as the initial cluster representatives that I'm showing here on the bottom. And then we go through, pick and assign based on the distance to that cluster point, the representative point, update the cluster point at each step, and repeat and repeat and repeat again until we have all our points assigned to our k=3 clusters in this case. Then once all the data is assigned to the clusters, we again sum the variation within each cluster here, and we get a value for that. Take all these horizontal lines, add them together, take that sum of within cluster variance, and you guessed it, repeat all over again in another iteration with new initial cluster representatives. So after several such iterations, k-means clustering will know which is the best result so far based on that value that we calculate, based on that smallest total variation within clusters value. However, it doesn't know if it's found the best result that it could possibly find, the best overall result. So it's going to perform several more iterations with different starting points and then pick the winner. So in this case where I've run these 4 iterations, the best answer is iteration 2. That's the one with the smallest value here of this within cluster variance. So that second answer, that second iteration, the answer that we got, is the one we're going to present to the user. Okay, so that was one dimension. What if we had more dimensions, such as 2 dimensions, like shown here in this classic kind of X and Y plot where we have our gray points to be clustered? So, well, just like before, we pick 3 k starting points, our k=3 in this case, so we pick 3 points at random and use these as our initial cluster representatives. Then we use the Euclidean distance. So in 2 dimensions, Euclidean distance is the same as good old Pythagoras theorem here, where we have the length of the side D here that we're after is equal to the square root of the lengths of the other 2 sides squared and summed, right? Or the square root of x squared plus y squared. In this case, that's going to give us that distance D between our gray point and the cluster representative. And we assign it to the one that's closest, in this case, the blue cluster. And we continue. We update the cluster center point for which we're going to measure subsequent distances, and continue and continue and continue for all points until we're all assigned, have all our points assigned to these clusters. And again, just like before, we have to use a number of different starting conditions before we know what's a good clustering answer in this case. What's the best answer we've seen over a number of different iterations is the one that we'll present back to the user. Okay, then what if we have more dimensions? For example, what if we have genes with counts here? So I've got genes down the rows, and I've got 3 different samples that I'm measuring this gene expression, for instance, in these 3 different cell lines or 3 different assay conditions, for example. How do we do this? Well, we could simply plot them, right? We could take those 3 samples and we could label them as X, Y, and Z and make a 3D plot. It would look maybe something like this, where the Z dimension is going off into the screen behind me here, and the smaller points are those that are further along that Z dimension. It's kind of hard to see it in 2 dimensions. And then we'd go through exactly the same procedure with initial cluster assignment at random, followed by distance calculations, updating the cluster representative, and continuing iterating. And we're going to use the same kind of distance, the same Euclidean distance, but in this case in 3 dimensions, where we have that Z as well as the X and Y squared there to sum up. So of course, we don't actually need to plot anything. We don't have to make some 3D plot. We can just use the distance, Euclidean distance calculation along whatever number of dimensions we've got, right? In this case 3, so we've got our X squared plus Y squared plus Z squared, all summed up, and the square root, that's going to give us our distance. But if we had more dimensions, we would do the same thing. So in R, we can do all of this is taken care of and done for us in one function call, and that's with Kmeans. That's the base R function for Kmeans clustering. It takes input X, which is your numeric matrix or data frame with your values in it, and it'll have 1 observation per row and 1 feature per column or variable, for example. And then the arguments here, centers, that's the number of clusters you're after. That's your K, what we typically call K in Kmeans. And then recall that Kmeans has this random component to it, and we can control the number of iterations, because the more iterations we run, the better. The more chance we will, the more improve the odds of finding the best model, if you will, if we run multiple times. So we're going to say restart please, in this case 20 times. We'll often pick a higher number here than the default, because it'll increase our chance of finding that best answer. So that's the Kmeans function. Again, X is our numeric input data, centers is the number of clusters you're after, and we're going to run multiple times with this nStart parameter. So if we run this on data that looks like this, and we say centers equals 3, so we're asking for a K equals 3 groupings, it's going to give a beautiful subgrouping of clusters like this. Clearly our eyes could do this, and Kmeans can do it too, where these are clearly separate and distinct, as we can see. Kmeans works well. So recall that Kmeans has this random component, and thus we run multiple iterations with different initial cluster centers by setting that nStart argument, that 3rd parameter that I showed in the previous slide. Well, in the case of the R function, the best outcome is based on the total within cluster, what we call sum of squares. How is this calculated? Well, for each cluster and for each observation, each point within that cluster, it determines the squared distance from observations to the cluster center, the center point of that grouping. And we sum all of those together to get the sum of squares distance. So here are some iteration examples where I've run Kmeans on this data set and got some different answers at different iteration points. And then the question is, which one's the best? Of course, I should have not used so many significant figures here, but nonetheless, the question is, which answer is the best out of these different answers that Kmeans has given us for this case? Well, here we would look at this sum of squares variance here, and we would pick the one with the smallest value. That's the one up here in the top right in this case. But notice something important here. I asked for centers equals 3 to give me 3 clusters, and that's the different colors in the different plots here. But by eye, we could probably tell that 2 clusters or 2 groupings would probably be a better choice here, right? But Kmeans will never give you an answer of 2 groupings when you ask for 3. Kmeans will just follow along obediently and give you your 3 clusters that you ask for, regardless of whether there could have been a better answer with a different number of clusters. In fact, it'll always give you what you ask for, right? So the warning here is that Kmeans will always give you the number of clusters you request, even if this doesn't make much sense. Okay, so it kind of imposes a structure on your data that you predefine in some respects. Okay, so then the question would be, well, how do we know what to ask for? What is a good number of clusters to ask for? So one common approach is to systematically try a range of different values and plot that value, return that sum of squares for the best answer you found at each k value. The so-called Scree plot can be produced by this, where we just plot that sum of squares value for each different number of k here along that horizontal axis. And then we look at this plot to try and find this point that I'm showing with the red arrow here, what some people call the elbow point, indicating where we've gained quite a bit in terms of the sum of squares for the fewest number of clusters that we're asking for. And this plot, people will often call this a Scree plot. And the name kind of, we mix metaphors with the elbow and the Scree here, but the name kind of comes from the fact that it's a bit like the Scree that would collect at the bottom of a cliff face. If you've got a natural outcropping of rocks or a cliff, say for example we walked from where we would normally have our classes together in Tattoo Hall, and we walked off towards the beach, towards La Jolla Shores or Black's Beach there, we would come to the cliff face, right? And there, if you take one more step, you're going to change in altitude quite dramatically. You're going to fall down that cliff. But at the bottom of the cliff, there's that point where there's all the Scree, all the rubble that is eroded off and it collects. This is the elbow point we're looking for. So if you took another step from that point, you get down onto the sand, onto the beach, and then you're taking steps, but you're not losing much in kind of altitude as you would go along before you get to the water. What we're really looking for is that point, that Scree point where the cliff face kind of ends and the Scree collects, because that's the point where you've got the most bang for your buck, where you've made the most drop in altitude, so to speak, for the fewest number of steps that you've taken, for the fewest number of k in this case. So that's the point that we're looking for. And we typically do this visually and then run the algorithm again with that k as our best answer. Okay, so your turn. I want you to open up R and try using Kmeans. And we're going to set k equals to 2 and then start equals to 20 in this case. And you're going to make up some data here, like I'm doing here. So here I'm making a little vector x, and I'm putting it together. So it's taking this R norm function, so it's making 30 points from a normal distribution that are centered at -3 and 30 points that are centered at +3, and putting them together. So we're going to have two clouds of two groupings of points, one centered at +3, -3, and another centered at -3, +3, and then we're going to plot them and see those points, and then we're going to try and answer these questions. Run Kmeans, inspect the results. What is the result object returned by Kmeans? How many points are in each cluster according to Kmeans? What component of your result object, that's the thing that R gives you back, tells you about the cluster size? That would be the number of members within each cluster. Which one tells you the cluster assignment or membership, so which points belong to which cluster, and how would you find out where the cluster center is? And what I want you to be able to do is to plot again x, but colored by the results of Kmeans, colored by the cluster assignment, and added cluster centers as blue points on it. Okay, so we'll do this in our hands-on section together as well, and I'll leave it here for now. And when we return, we'll dig into other clustering methods and other dimensionality reduction approaches. So I'll see you then. Thanks. I'll leave this up.